I think that all of us here can agree that science is pretty cool. But there's a lot of different fields of science. In something like chemistry, we don't exactly think about our reagents feeling any particular kind of way about being mixed up in our glasses. But we cannot say that same thing for every field of science. When you get more biological, well, we have to get biomass. I would argue that some of that biomass would rather not be used for our experiments. For example, something like fetal bovine serum, which is used in cell culture, is not made by us in lab. The fetal bovines make it, and then we take it from them, which also includes taking the life of the cow. Actually, about 1.8 million annually just for fetal bovine serum. While I love a good burger as much as any American, why don't we use an organism that has made its juices specifically to be eaten? This is where our star, the humble peach, comes into shine. Fruits have sugar because the plant wants us to spread their seeds. So why don't we borrow some of those sugars that they've already kindly made for us to make some biomaterials that are compatible with our own bodies and sustainable? We can use peaches to create a renewable bio-ink that is sustainable and decreases the need to use animal or petrochemical biopolymers.
Pectin is already a chemical that we produce commercially. We produce it from apples, but we could also use peaches because they come from the same family. Pectin can be found throughout the fruit tissue, particularly in the middle lamella and primary cell walls, where it exists as protopectin. First, we must extract the protopectin using a hot dilute acid, like citric acid, which will hydrolyze the protopectin into soluble pectin built mainly of galacturonic acid units. For the best control over the degree of esterification, we add a saponification step where a base lowers the degree of esterification. Next, we blend this pectin with tempo-oxidized cellulose nanofibers, another biomass-derived material that can be obtained from various plant sources including agricultural waste. These nanofibers help create an ink that can withstand being extruded and withstand stress similar to early cell morphogenesis. Its biocompatibility makes it a perfect candidate to be used to make personalized 3D scaffolds that mimic the cell's natural extracellular environment.
Biomedical engineers, biochemists, and many others need a sustainable bioink. Our other options we have now are incredibly cool, like decellularized tissues or blending up some poor creature and extracting their collagen, but the thing about these methods are they are incredibly expensive and super time consuming. But the wonderful peach has already created a great sugar just for us, and also every other material we need for this bioink. So, to help the environment and the sanity and pocketbooks of the scientists we work with, we should use the sources that want us to use them.